So I will give a talk about the work that we do at ProteoMediX, which is that we developed a Luminex-based diagnostic test to reduce unneeded prostate biopsies and to improve the detection of prostate cancer. First, I will give you a short introduction about prostate cancer. Then we will go into the Luminex assay development that we did, and of course some clinical testing that we performed with those assays. And at the end, a short summary and conclusion. So first of all, to give you an idea of the importance of prostate cancer, here you see the number of new cases in the US 2017. And as you can see, prostate cancer is the most frequently diagnosed cancer in men. About 1 in 7 will be diagnosed with prostate cancer in his lifetime, and 1 in 39 men will die of it. Now, currently there are several unmet clinical needs in prostate cancer. When it comes to diagnosis, there are certain tools already existing, but many of them are inaccurate, and it ends up with more than 60% of all biopsies that are performed being negative. When it comes to prognosis, this is basically missing. So 2/3 of the prostate cancers are low grade, but still nowadays often prostatectomy is performed, so the prostate is removed, which is a huge overtreatment. And for treatment, it would be nice to have, since it's quite unspecific nowadays, to have a test that can tell us whether a patient will respond to a certain drug or not. So, as I said, nowadays we have overdiagnosis and overtreatment, which results, of course, in huge costs for the healthcare systems and also in unnecessary procedures for the patients. So the whole story started with the doctoral thesis of Ralph Schiess, co-founder and CEO of ProteoMediX, who started with generating a P10 cancer mouse model by performing a conditional knockout of this protein, which is involved in a signaling pathway commonly mutated in many cancers. Then wild type and these knockout mice were compared by quantitatively measuring the protein levels of a huge panel of proteins and comparing whether we can see some differences. This ended up with a panel of candidate biomarkers, which were then verified in humans, autolog proteins that are existing in humans, and both in serum and also in extractions of prostate tissue. So we ended up with a final panel of markers to further investigate. And what we can see here now, basically those proteins that we found are not just random proteins involved in any function that doesn't have anything to do with cancer, but they are actually all involved in the hallmarks of cancer stated by Hanahan and Weinberg. Aspirin, for example, plays a role in evading growth suppressors. 2 other proteins are involved in activating invasion and metastasis, and we also have proteins that play a role for resisting cell death and inducing angiogenesis. Now, a short summary of all this. We started with this cancer mouse model, verified it in humans, and received a panel of many protein biomarkers that we can use basically to develop 3 different kits. The first one, the diagnosis kit, should reduce the many false positive results that we still have nowadays. The prognosis test is intended to assess the aggressiveness of the disease and to help in taking a decision how to treat it. And a prediction assay should stratify patients for a response to a therapy, so that we can so that we know already whether treating a patient with a certain drug will give a result or not. So we have those 3 products in the pipeline, and in general, all of them start with the mass spectrometry technology, where we find the initial proteins to follow follow up with. Then we generate a functional prototype and validate those markers in a pilot study. Afterwards, we generate an advanced prototype. I will go into the details about the differences later, and conduct further clinical studies. And at the end, we really produce an IVD-ready assay, CE mark, and conduct the final pivotal study before it can go on to the market. The diagnosis kit is already quite advanced. We have the advanced prototype, already performed some studies on it. The prognosis test currently is at the functional prototype stage, and we already performed also some studies. And the third product for the treatment selection is still at the end of the mass spectrometry phase. So, about the Luminex assay development, most interesting part probably. Here is a small overview of the kind of dimensions and, that we are capable of looking at over the different phases. So with the initial biomarkers that we investigate with mass spectrometry, there we can, of course, look at many candidates, thousands of different candidates, but as the technology is not really suitable for high throughput, we can only look at small study size or cohort sizes lower than 200. The functional prototype, which is a Luminex assay then, that we develop ourselves, there we can look at approximately 10 candidates, still quite a lot. If we, of course, what we then do, go up with the cohort sizes to 200 to 500. The advanced prototype is already with a bit less candidates, as we filter out some that are not performing too well. And we also go up with the cohort sizes, which takes then a bit of, more time. And at the end, we transfer everything to ELISA, the final biomarkers, and there, of course, we can increase the study sizes again. So for the functional prototype, the Luminex platform is actually really an ideal thing to work with. We have 2 MACPIX instruments, and we saw 5 points that are really an advantage of using this technology over other immunoassay technologies, as for example, ELISA. One of it is a short development time. With a bit of knowledge, what to look at and how to develop an assay, we are ready in 1 to 2 weeks to measure clinical samples. We need really low amounts of reagents, which reduces also the costs. We could break it down to really having 100 microgram of antibodies and 100 nanogram of protein. An amount that might be intended to measure maybe 5 microtiter plates, but by transferring it from an ELISA kit that we buy usually, so for the functional prototype we buy commercial reagents, we can extend this by transferring it to Luminex to 20 to 60 plates, which of course gives us the capability of measuring many more samples. The signal range is extremely broad in Luminex, which is also a nice thing. If we have protein biomarkers that are spread over even 3 concentration logs, we can dilute all the clinical samples at the same dilution and still measure all of them without any problems of sensitivity that they are below the limits of quantification or above. The assays are very robust, so we have very low signal variation and also very low intra and inter-CVs of usually around 5 to 10%, certainly below 15%. The linearity of dilution is also easily achievable. And last but not least, very important for a startup, we can save a lot of precious sample material. They are expensive, they are hard to get, especially for a new company. And by reducing the assay volume to 50 microliter in half area plates, we managed to save a lot of material. And also, usually we need to dilute quite a lot. And we have a panel of different assays, so we can go on from the first pre-dilution and save a lot of material. With 10 microliters, we can measure maybe 10 assays even. With this functional prototype, we conduct a pilot study to filter out the markers that that we cannot transfer from mass spectrometry basically to the immunoassay format. And for those markers, we generate our own reagents by immunizing. So we receive a panel of maybe, usually it's around 5 to 20 antibodies, but we have no clue yet which ones work best together. So here we can really use the strength of the Luminex system by multiplexing. We couple every antibody to a different bead region, then mix all those bead regions together and screen it against, again, every antibody to find the best capture detection pair. And this massively reduces the reagent consumption and also the time that we need to find the ideal pair. Plus, on top, since we already have the reagents from the pilot study that we conducted previously, we can directly compare the results that we get with our own reagents to the ones that we measured, not to lose this link. And then afterwards, we go into the assay development with our pair that we chose. Usually, here we test a little bit more, so it takes us 2 to 4 weeks to have everything set for the next clinical study, which is not, what is nice here, again, the low reagent consumption. Antibody and protein is usually not limiting. And that's great because for a first production, it might be that the yields are actually not quite high, as this can be still optimized. But that's not a problem with Luminex. But however, we do singleplex assays, for various reasons. So here, a principle of our two-step Luminix assay. We do, at the beginning, we had a 3-step, actually, but then we realized that we can just mix everything together, so really the bead-coupled capture, detection, standard QC sample, one of them, in one mix, then incubate for an hour at 37 degrees, afterwards 30 minutes with streptavidin fucoerythrin, and then we can already measure it. And this simple format gives reliable results in a short time, which is of course great. So to the clinical testing. Now, our aim is to improve the prostate cancer diagnosis for the first kit that we want to bring onto the market, to reduce the many unnecessary biopsies, and by this provide a solution for this prostate cancer overdiagnosis that exists. Now there are already diagnostic tools on the market, but what differs our kit from those is the higher accuracy, which you need desperately to be able to achieve this goal of reducing unnecessary biopsies. It's a blood-based test, so it's non-invasive and it's highly reproducible, and it's easy to automate and can be adapted to clinical practice. The current clinical practice looks like this, that the PSA blood test is performed. PSA is a prostate-specific antigen. In combination with a rectal examination, men fall into one of 3 groups. Either everything is normal or abnormal. But then there is also this gray zone, and that's a great problem that we have nowadays. If we have an intermediate PSA of 2 to 10 nanograms, but a negative DRE, nothing suspicious there, and an enlarged prostate, which is quite normal for elder men, all those men ending up in this gray zone undergo prostate biopsy, which is an invasive procedure and can also lead to inflammation or other problems. And 60% of all those biopsies are negative nowadays. So we would like to bring in a second test after the PSA and DRE combination. And if our test is positive, then men can still undergo prostate biopsy to verify that they really have cancer, or whether they really have cancer. But otherwise, they can go into active surveillance, remeasuring or redoing the test after a certain time. By this, we aim to reduce the negative biopsies by 50% or more. So we conducted the first, or this was actually the second clinical study with around 260 patients, with patients from 2 clinical centers. So first we did a univariate analysis of all the individual proteins, and what you can see here is are the concentration levels for prostate biopsy negative and positive men. And as you can see, not all levels are different between those groups. So that's one thing. As I mentioned before, we cannot always transfer what we measured in mass spectrometry to the immunoassay. However, percent free PSA and thrombosponin, thrombosponin is one of our markers, show really a good discrimination power between biopsy positive and negative. And then we did a ROC curve analysis on different models that we created on this data set. And here you can also see, most important is the jump from the blue curve. Maybe I should explain this graph quickly. On the y-axis we have the sensitivity, which gives us an idea about the fraction of true positive findings compared to true positive and false negative. So usually we want to have not more than 10% false negative results and still have a good specificity on the x-axis, meaning that we don't get too many of false, it's always confusing me with this figure. We don't want to find too many false positives, right? That's how it is. Sorry. And as we can see here, the red curve, you always have to look at the area under the curve for those, is quite good. And this is a combination of already available information. The percent free PSA is usually available, the age of course too, and two of our protein biomarkers, cathepsin D and thrombosponin, which is a quite good result. Then we conducted another validation study with more than 470 patients. And as you can see here, we could basically get the same result again with a similar panel, percent free PSA, cathepsin D, and thrombosponin. And by this, at 90% sensitivity, which means 10% false negatives, we are able to reduce unnecessary biopsies or negative biopsies quite significantly compared to just free PSA alone, which is currently in clinical practice. And in combination with this, we can even achieve 60% reduction. So this is a quick summary of all the 3 studies that we conducted here. And you can see that the results are actually quite similar. The number of negative biopsies compared to the number of negative biopsies, if we would use the ProteoMediX diagnosis test, can be reduced by more than 50%. What are the components of our kit? It's basically this total and free PSA, which is already measured as a standard nowadays. And then, as I mentioned, the 2 prostate cancer proteins that we have in our panel. We then calculate with an algorithm that we generated a PMX score, which indicates the risk of having a positive biopsy. And this score aids us in taking the decision whether a biopsy should be performed or not. So, quick summary and conclusion. Currently, for prostate cancer diagnosis, there is a large unmet clinical need for better diagnostic tools. The Luminex platform was for us an ideal tool to get good assays in a short time. They have a large range. The assays are very robust, and it saves a lot of reagents and samples. So it's an ideal tool to make quick progress at an early phase. The final PMX diagnosis test will provide a solution for prostate cancer overdiagnosis. And then an outlook and our last activities. What we did now, we transferred those Luminex assays to ELISA for IVD kit manufacturing. And with this kit, hopefully we will conduct a prospective multi-center study with 6 centers. So I want to thank all the involved contributors. First of all, the Martini Clinic and the Cantonal Hospitals and Gallen, who helped us in conducting those clinical studies. The NMI, which was a great aid at the beginning, developing the Luminex assays, and of course ProteoMediX, all the involved players, Annalisa Macagno, the head of our assay development team, my colleagues in the lab, Alcibiade Attanasio and Anja Wittig, and then also Bruno Golding, the head of our clinical development, and Ralf Schiess, the CEO.